So I have the great pleasure of being able to be not only the first speaker here, but have the opportunity to give you a sort of ovarian cancer 101. I understand that the amount of information you know about ovarian cancer already is going to be quite variable. Some of you, this will be a lot of new information. Some of you might already be familiar with some of the information I'll present. What I'd like to do is sort of set the stage for the following two speakers who will give you much more detail about things that I think you'll want to know. And so the topic is ovarian cancer 101 and the impact of research. I'll start with cancer generally. Everyone knows maybe that cancer is a malignant growth that spreads, unfortunately, to organs that are really important for our survival. And 85,000 Canadians will die of cancer this year. So it is a huge health problem. These numbers are actually taken from the American Cancer Society because they had lovely figures I could pilfer. But the percentages are the same in Canada. So you see percentages of men who die of different kinds of cancer on the left, and on the right you see women. And so you see ovary there is the fifth ranked cause of death by cancer in women. 5% of women who die of cancer die of ovarian cancer. And so if you look at the incidence doesn't even rank in the top few types of cancers in women. But if you look at the ovary, that's not going to work. Okay, so if you look at the ovary, which is the burgundy line here, you can see that it's essentially flat, which means this is the number of women who die every year from ovarian cancer, and that hasn't gone down. It hasn't gone up, but it hasn't gone down, and clearly that's what we're trying to achieve by research, is to make that line go down so fewer women die of ovarian cancer. And so I'll throw a few sciencey slides in once in a while. I know most of you are not scientists, so I'll try to keep it light. But the big question is why did I get this cancer? Why do people get cancer, generally speaking? And probably the easiest answer is mutations. We all have cells that make up our body, and inside those cells are DNA. And the DNA is what dictates the health of the cells and the whole body. And so if a mutation occurs in the DNA, this is the structure of DNA, if a mutation occurs, that's going to change potentially how the cell behaves. And so when it can do nothing, you might have a mutation that will get repaired, and so the mutation goes away. And sometimes a mutation has no effect on the cell, so it can stay, nobody fixes it. But as we get older, our cells get less able to fix those mutations, and so they tend to stay, and they tend to accumulate, and so that's why cancer of all types actually increases with age, is because our cells are getting really bad or is not as efficient as correcting those mutations. And so the cells with those mutations then can accumulate, and at some point or another, the body will not be able to handle all those mutations, and the cells will grow. And if you look at it in this way, if this is a normal layer of cells in your body, it could be your skin, for example, and normally there's a balance between when a cell decides to divide, and this is how we keep our bodies fresh and young, is our cells divide, but some also will die. And so this is a cell that's chosen to divide, and we have a balance of cell death genes that will cause some of those cells to divide or to die. And so you end up with a balance so that you don't end up with a lot more skin than you need. You have some renewal of cells, and you have some cell death, and there's always a balance in the ideal world. Cancer cells, however, throws that balance off. And so here you have the same layer of cells, and whether it's because of cigarette smoke or UV light or pollutants or bad luck or whatever risk factors might have caused a mutation in a cell, and that mutation, let's say, will allow the cell death gene to be mutated, so it can no longer function. And so what happens is that the cell will get the prompts to divide like it normally does, and it will divide and divide and divide, but the cell death gene is not going to be activated, and so it can't cause that balance to be restored. And so you just have that cell growing and growing. And so you end up with what are called immortalized cells, and eventually they will form a tumor. So essentially a cancer is just an imbalance of cells deciding to divide or deciding to die, and usually that balance is working pretty well in most people, in most cells. But every once in a while, if a mutation happens to occur in a cell death gene, the balance is thrown off. Make sense? Okay, good. Okay, so what are the challenges particular to ovarian cancer? And so this is the fact sheet. This is the stuff nobody wants to see, but this is the stuff I have to show you just to sort of lay the groundwork with what are researchers and physicians dealing with when we're talking about ovarian cancer. Well, there's a lifetime risk for women of 1 in 70 women will develop ovarian cancer. So that's actually a pretty low incidence rate compared to many other types of cancers. It's about 28,000 new cases in North America. Because Canada has a population about 1/10 that, we're talking about 2,800 new cases in Canada each year. The average age at detection is between 56 and 60 years of age, and about up to 20% of the cases we now know are hereditary. Most of those are associated with mutations in BRCA1 or BRCA2, and we'll talk about that in a minute. And then the rest are what are called sporadic. No family history, and all of a sudden a woman is diagnosed with ovarian cancer. It's like, where did that come from? And that's a non-genetic sort of origin, and that's sporadic ovarian cancer. As most of you probably know, the symptoms are often very subtle. So subtle that women will too easily dismiss them, and that is part of the problem. And we don't have a reliable screening test for early disease, and I'll talk about that as well in a bit. And so about 60% of cases are fatal, but 40% of cases are cured. So they're just keep that in balance. But the ones that are fatal tend to be, unfortunately, a little too quick for my liking. Most women will die within about 3 to 5 years. The only thing that has improved in the past 30 years since I've been doing this work is that the longer-term survival. There are more women living to 5 years and beyond than ever before. And so that has been sort of the big advance in terms of the survival of women, and it's still not good enough. When I started this work in the early 1990s, there were, I could count 3 scientists in Canada that were doing research on ovarian cancer. Nellie Auersberg in Vancouver, Steve Nayrod in Toronto, and Anne-Marie Mesmasson in Montreal. And I thought, well, this is, no wonder the survival doesn't change. There's nobody in Canada doing research. And these are all very well-recognized and high-profile investigators. But that's all there really was. And so that's one of the big issues, is that you have to have a research community if you actually want to change what's going on in the care of these patients and the treatment of these patients. And so where we started very small, I don't have to dwell on this because Elizabeth has already pointed out that one of the first things that Ovarian Cancer Canada did with the investment that it had was to establish an endowed chair in ovarian cancer research in Ottawa, at the University of Ottawa, and I am that person. I've held that position. I just was renewed after 15 years for another 5 years, so I must be doing something right. And so what I said earlier still holds true. I didn't want to play alone in the sandbox. Very good friends with Nellie Auersberg and Anne-Marie Mesmasson, wanted to connect right away, but we weren't gonna, with just the 4 of us, we weren't going to be able to change the world that we wanted to change. And so that's where I started this conference, and Elizabeth pilfered my slide, but so this is where we are right now. Just down the hall, there's 160 ovarian cancer researchers having this meeting. Went from 3 people that were doing research to, in Canada, there's more than 60 ovarian cancer researchers now. So that is the part that makes me the most, I get goosebumps when I talk about this because, you know, it's not that, I just got the ball rolling and I don't like to play by myself. So all I did was start to bring people together. And as Elizabeth pointed out, training was a big part of that. And so the people who have gone to this conference, who have gone to the trainee forum, are now ending up in like last week I found out that one of my previous graduate students just got a job as a professor at Harvard for an ovarian cancer program, which makes me very proud and knows that this is working, right? This is where, it doesn't matter where they're working. Harvard is a pretty decent university. So even if it's not Canada, at least we are building the ovarian cancer capacity in North America. And so what can we do? What have we done? Have we done anything? And so the way I've worked this is that the rest of my talk are going to be questions that I hear all the time. And what we're going to try to do is answer some of them. And some of them we don't have all the answers, but we might have some clue. And what everyone needs to know is what are the risk factors. We need to identify what it is that's going to put us at risk. And there's 3 categories for that. The first is genetic risk. I've alluded to this already. BRCA1 mutation or BRCA2 mutation. These genes are called tumor suppressors, meaning that when they're there and healthy and normal, what they do is suppress tumor formation. So critically important for that cell to stay quiet and only divide and die when it needs to, but it suppresses the formation of tumors. But women who have a mutation in BRCA1, right, so it's not functioning properly anymore, have a lifetime risk of getting ovarian cancer of 35 to 70%. So that's pretty high. Of every 100 women, 35 to 70, depending on the type of mutation that they get, have a risk of getting that cancer. So that's a lot higher than the general population. General population is less than 2% of women will get ovarian cancer. The BRCA2 mutation is not as aggressive, but it will still, for every 100 women who have a BRCA2 mutation, between 10 and 30% will actually develop ovarian cancer. And again, where that mutation is in the BRCA1 or 2 gene says a lot about what the risk is going to be. And BRCA mutations are about 10 times more common in certain populations. Ashkenazi Jewish women have a higher propensity to have BRCA1 mutations, and there's a French-Canadian population that also has risks of BRCA1 and 2 mutations. So there are certain ethnicities that have a higher risk, and of course they are monitored more carefully as well. There is another syndrome it used to be called Lynch syndrome. It's now called hereditary non-polyposis colon cancer. So the higher risk is for colon cancer, but it has mutations. There's a number of genes that are involved in keeping DNA safe, repairing those mutations. That's really important for it. We have genes that repair those mutations, and as long as those genes are functioning well, we can fix mutations that occur. In HNPCC, those mutations are not fixed very well, so you accumulate mutations, and that puts an increased risk, primarily for colon cancer. But about 1% of all ovarian cancers actually are associated with having a mutation in a gene associated with DNA repair. There are others that are less common and that are other kind of gene families that we're beginning to identify. When you put them all together, BRCA1 mutation, 2 mutation, the mutations associated with this family and some other smaller ones, it's creeping up. Between 15 and 20% now are associated with some mutation that we can identify. Another area of risk factor seems to be related to reproduction. Of course, the ovaries are critical for reproduction, and so a lot of the things that the ovaries do or naturally do seem to be associated with risk, which is unfortunate. So there's a history of infertility, or prolonged use of fertility drugs seem to increase the risk modestly, and particularly for what are called low malignant potential tumors, LMP tumors. So it is a slightly increased risk. Many studies are out there saying there's no increased risk, so it's not a very strong risk factor. Nulliparity, which means never having any children, that is a risk factor. Tubal ligation is actually protective against ovarian cancer for reasons that we actually really don't understand yet, but it seems that women who have tubal ligations actually are quite well protected against ovarian cancer. A risk factor is estrogen replacement therapy, so postmenopausal women who don't handle or have really dramatic responses to their loss of estrogen at menopause can take estrogen replacement therapy. That does increase the risk of ovarian cancer. Not hugely, very small increase in risk. If you take estrogen replacement therapy with a progesterone at the same time, it seems to be that the estrogen risk is reduced and it's not so different. So estrogen alone is more risk factor than the combination estrogen and progesterone replacement therapy. And we now know there's evidence that maybe even endometriosis can be a risk factor for certain subtypes of ovarian cancer. And so if you look at these factors, then it will make sense that the two factors we know that most reduce the risk that women have control over are use of oral contraceptives and full-term pregnancies, right? So I said up here, the nulliparity is a risk factor. Well, actually, full-term pregnancies and the use of contraceptives both reduce the risk, and actually quite substantially. They're among the biggest factors to be able to reduce cancer risk of any type of cancer. So use of oral contraceptives has a little bit of an increased risk for breast cancer, but what it does is it stops ovulation. And ovulation, or the number of ovulation a woman has in her lifetime, seems to be associated with a higher risk of ovarian cancer. And we actually don't know why. That's one of the things my lab is working very hard on, is why is ovulation a risk factor? But it makes sense if you look at this. Oral contraceptives, that's what they do, is they stop ovulations. And for every year that a woman is on oral contraceptives, the risk is reduced by 10%. So a woman who's been on the pill for 5 years actually has about half the risk of ovarian cancer as the general population. That's pretty substantial. And the same thing with full-term pregnancies. The more full-term pregnancies you have, the lower your risk of ovarian cancer. It never goes down to zero. That risk is always there. But it's dramatically reduced by those two factors. And so the other category, the final category, the odds and ends category, age. As I mentioned earlier, we do accumulate mutations in our cells over time, and our cells are less able to get rid of those mutations. And so about half of all ovarian cancers are found in women over the age of 63. There are some studies that would suggest that obesity would increase ovarian cancer. That's actually a very slight increase. Smoking may increase the risk for one type called the mucinous subtype of ovarian cancer. Being tall, which is fortunately not my problem, but that actually has been associated with a slight increased risk as well. These are things we really can't do much about. And this is the one that has hit the news, and so I threw a few pieces of information about there. What about talcum powder? So I don't know how to answer. I do not know how to address what's going on in the United States with Johnson & Johnson being sued by women who claim to have used talcum powder in their past. And the story is really unclear at this moment. I haven't figured out how it's processed, and maybe you'll have more information about that than I will. But so talcum powder has historically been used by women to sort of keep themselves fresh down in the nether regions. So if they're applied to the genital area in your underpants or sanitary napkins, it was an attempt to keep moisture at bay. Historically, before the 70s, talcum powder had asbestos in it. So that put women at risk, and then they realized asbestos is not a good thing. So in the 70s, they took asbestos out of talcum powder. And many are now cornstarch based. So the earlier studies would indicate, yes, there's a modest increase in risk for ovarian cancer if you use talcum powder down in that area. And it's likely associated to the use during the 60s and early 70s when maybe asbestos was in that talcum powder. They were always reliant on women's memory. They have ovarian cancer in 2010. Oh yes, I used talcum powder back in 1960 when I was young. The reliability of the memory, what kind of talcum powder they used, how often they used it, there was that increased risk, but it was always reliant on remembering how often you use talcum powder. There are 2 prospective cohort studies, meaning that women are starting at the study and documenting their use of talcum powder. And both of those studies then are probably more reliable, and both of them found no increased risk. So from what I can see from the literature, and being a scientist, that's really where I get most of my information, it would seem to be that anyone using talcum powder from the 70s on probably doesn't have an increased risk of ovarian cancer. However, we have 2 settlements in the States in the last few months where huge amounts of money are given to women who indicate that their ovarian cancer likely came from their use of talc. I can't mesh those 2 ideas. It doesn't agree with what I see in the literature. Maybe they have access to information that I don't have. Maybe it's the American way to have these lawsuits come forward and then eventually be settled for something less. Johnson & Johnson does admit that the earlier talc that they had was the form that had asbestos in it, but that changed in the 70s. So I can't give you a clear answer on this. All I can do is say if you look at the studies that I see in the literature now, it doesn't look like talc produces an increased risk for women who are using it now. Having said that, every website you go to now is advising that you use cornstarch. If you want to use something to keep yourself dry and fresh, that cornstarch works just as well, and there hasn't been any indication yet that that's an increased risk for ovarian cancer. However, no one's done the studies yet to actually prove that. So I'm not going to advise you one way or the other. You, like a scientist, I give you the information. You can do with it what you will. But that's what I know about the use of talc. Okay, so the rest of the talk is going to be 5 things we know now that we didn't know 10 years ago. And these are based largely on the kind of questions that I get when I talk to audiences. And so the first one is that some ovarian cancers start in the fallopian tube. And I write some because there's huge debate of how many is some. We always thought historically that this is the ovary here and that this is, it's called ovarian cancer for a reason, right? It must start in the ovary. But in the past decade or so, we've learned that some of the cancers that are called ovarian cancer probably start in these little finger-like structures here that are called fimbria. For those of you who aren't familiar with female anatomy, this would be the uterus, this is the fallopian tube, and this is the end of the fallopian tube, and that's the distal fallopian tube or the fimbria. And so when women, you've heard of Angelina Jolie, she, gets a lot of PR, which is good because it promotes awareness of how you can prevent or try to reduce your risk of ovarian cancer, in her case, breast cancer as well. And so she had her ovaries and the distal end of the fimbria removed to try to reduce her risk of ovarian cancer. Because we know that some come from here, some likely come from there, and the only way right now that we can prevent this disease, reduce the risk, is to take both of those out. So not the ideal strategy for prevention, but that is what happens. This is what the real anatomy looks like. There's the ovary, and then lying over top of it, you can see the fallopian tube. And so, as I said, historically, we thought that the ovary was the source of the ovarian cancers, and largely because at ovulation, which is one of the risk factors, the surface has to open up so that the egg can leave the ovary, and well, then it's got to fix itself. And we know that in the body, areas where there's a lot of wounds and repair and wounds and repair are the areas that are prime for developing cancer. So this made sense, scientifically, that every time you ovulate, there's a wound that needs to be repaired, and that repeated wound repair was causing an increased risk for ovarian cancer. But then Angelina Jolie and others, women who are at high risk because of BRCA1 or BRCA mutations, started having their fimbria, their distal, the fallopian tube and the ovary removed. And pathologists began looking very carefully at the fallopian tube and found that there were early cancers in those fimbria. And it was like, well, we don't have a high risk of fallopian tube cancer that we're aware of. And so they put two and two together and realized that about half the time that a high-grade serous cancer is diagnosed, they can go back now and look at the fallopian tube and see about half the time you can see early cancer in the fallopian tube. So we know at least half of what's called high-grade serous cancer probably comes from the fallopian tube. And there are some in the country who think that all high-grade cancers probably come from the fallopian tube. We don't have that answer quite yet, but we know that at least half do. Okay, so that means that the recommendation, particularly if you're from BC, because this is really strong, they made it a provincial recommendation, is that women should have their tubes removed if they're having abdominal surgery for any reason. If you're going to have your abdomen opened up or your pelvis opened up for any surgery, the recommendation has been to have your fallopian tubes removed at the same time, assuming you're past your reproductive years. You really don't need them if you're not going to reproduce, and if that's really where ovarian cancer comes from, this might be a nice incidental way of removing the tissue that is most likely to develop the disease. And so it started in BC and it's been rolling across the country. I know we do it in Ontario. We just had a meeting with gynae oncologists a couple of weeks ago at Ottawa, and I learned that a good number of them are doing what I call prophylactic salpingectomy, removing the fallopian tubes to try to reduce the risk of ovarian cancer. And as I said, this is not a good strategy, right? For having surgery to have body parts removed is not a good strategy for cancer prevention in any context, but it is one that works. And so this is the one that is currently in place while we try to figure out another way to prevent the disease. This is the other question I get all the time. Why is it taking so long to develop a test for early detection? And wouldn't it be wonderful if we had an answer? But it's not for lack of trying. I think the most important thing I can impress upon you is that there have been 2 massive studies done in the United States and in the United Kingdom. For example, in the United Kingdom, that study included 202,638 women. This is not a small clinical trial. These are massive studies, and the one in the United States is also very large. And so they were taking the factors that we use to try to diagnose ovarian cancer and see if we can use that as a screening tool. Can we measure blood CA-125 every year and then do an ultrasound if the CA-125 levels look a little high? And then if the ultrasound looks like there's something unusual, we go in and do surgery to see if there's anything there. And so the disappointing thing about both trials, the other one, the Lancet published the study just a couple months ago. They had 11 years of follow-up on all those 202,000 women. And both of the studies generally have found that you can find more cancer in women who are screened. But it had no impact on whether the women will die from that disease. What happened in the United States study is that you diagnosed more, but you ended up doing more surgery on women, and there was as much risk associated with doing surgery, because it's not an innocuous thing like taking a pill. This is surgery. And so there were problems occurring during the surgeries, and that caused even greater risk to some of these women. And so, but the overall mortality rate was not different if you detected the disease early. The UK study has a little bit of a positive element to that report, and that it said that if you monitor women for 7 to 10 years, you might be able to see a decrease in mortality. Unfortunately, that's where we stand right now after 11 years of follow-up. We're at that point where it's, okay, maybe if we monitor them for up to 10 years, there might be showing a decrease in mortality. So we're going to have to wait another 3, 4, 5 years to see whether that actually is true. But it was, I think, a 15% decrease in mortality by screening hundreds of thousands of women. So that could be important, but of course somebody's got to figure out, how do we get women on an annual screening program? Who's going to pay for that? And is it really going to change whether women are going to be saved from that process? So I have to tell you that in the research world, this was about the most devastating piece of news that we could get from both the American and the British studies, because we had all put our hopes on these two massive studies that this was going to tell us what is the best way we can screen for ovarian cancer. And it doesn't look like either of them yet are going to be good enough. And it's going to be a long time before anybody can mount a study of this size again with another different idea. These were the best candidates we had. So it really is quite disappointing that we're at this point with these two large studies done, and really we have nothing to show for it yet to say that we have a screening test that's reliable or will protect the lives of women. So it's not, as I said, for lack of effort. It's that the screening methods that we've chosen to study are not going to likely work. Okay, so what do we do in that context? Well, Elizabeth pointed out that 90% of women with ovarian cancer who were surveyed a few years ago found that they had at least one symptom that had been associated with that cancer that they knew about, but they maybe didn't act on right away, or they just dismissed it because it was something that's a little bit of bloating. Well, we've all gone through that thousands of times. I love this cartoon from Ben Wicks. Forget the tall, dark, and handsome stuff, which is hard to do if you've met Justin Trudeau, really. But forget the tall, dark, and handsome stuff. Any signs of ovarian cancer? And so, what do we watch for? Well, if we can't screen for it, then women need to be aware of the symptoms. And the top ones are the ones that are most commonly associated with ovarian cancer. I'm sure some of you are familiar with those symptoms because they're things that women experience all the time. These are sort of less common, but also symptoms associated with ovarian cancer. And the key to all of this is, are the symptoms new? Are they persistent? And are they frequent? And so if they persist for 2 to 3 weeks, if they are new, you've never had this symptom before, or it's staying and it's not going away like it normally does, those are trigger factors that say you need to go see your GP. You need to talk, and you need to say, might this be ovarian cancer? Because I have these symptoms and I don't know what they are. I'm eating well and everything is fine, but I have a lot of pain in my lower back or my abdomen that I've not had before. Can you please check this out? So you've got to bring it to the attention of the GP and raise the issue, could this be ovarian cancer? Because they might not think of ovarian cancer as the first option. Okay, so what does early ovarian cancer look like? If we can't screen for it, can we find a way to at least diagnose it early? And so my lab has been spending a fair bit of time trying to see, okay, what are the origins of ovarian cancer? And so if it was like this, we'd have no problem at all saying, oh yes, that's an ovarian cancer. Of course, what ovarian cancer, right, is embedded in the ovaries, which are embedded someplace in your abdomen, and you really can't feel them unless the bloating or something, or the tumors get large, and then already it's metastasized. So we need to figure out what early ovarian cancer looks like while it's still in the ovaries or the fallopian tube. And so we've been doing that with mouse models. And so if there's any PETA fans in here, don't shoot me yet, because there's a good rationale for this. We can't study early ovarian cancer in women. Most of women who are diagnosed at stage 3 or 4 when the disease is already spread throughout the pelvis or even the abdomen. I don't know where the tumor began when you have tumors everywhere, and neither does a physician. And so the only thing we can do is try to model this in a system where we can study normal transitioning to cancer. And we use mice. Most of the labs that do study the origins of ovarian cancer, we use mice, because we can create a mouse that actually has ovarian cancer that looks exactly the same as the kind of cancer that some of you have had. And so I've spent the past decade creating all kinds of mouse models for ovarian cancer and trying to model the different types of ovarian cancer that we have. And this was the very, very first transgenic mouse model of ovarian cancer. I worked with a lab in Philadelphia, Tom Hamilton, and together we created the very first mouse model of ovarian cancer. And now this looks weird to you. I'm sure you have no idea what it is because you're not looking at mouse reproductive tracts all the time. So this is the cervix down here. This is the uterus. And in the mouse, these are the fallopian tubes, or we call them oviducts. And then this little dotted line indicates about the size of the normal ovary. So right away, what you can see is that we've got two massive bilateral tumors in this mouse. I can't tell you all the moments of excitement in my entire career. This one was at the top of the list. This was the very first model of ovarian cancer where normal became cancer in an animal system where we could study it. And so it's been used quite extensively. We're using it to test all kinds of new cancer treatments. Some of them are oncolytic viruses with my colleague John Bell. I'm not going to say much more about that because another speaker is going to address that. And we've also used it to try to see how do the ovarian cancers begin. This is critical. And so I'm just going to give you two slides here. This is a normal ovary from a mouse. That's actually an oocyte or an egg. And what you see here are the epithelial cells, or a layer of cells that sit on the surface of the ovary. And all the brown ones are the ones that we've induced with an oncogene, a cancer-causing gene, and we're sitting there after 65 days, 2 months. We activated the oncogene 2 months ago, and they're still sitting quietly on the ovary. So that's pretty good. Like, it's not making cancer right away. 1 month later, if you look very carefully, you start to see little tumors growing on the surface of the ovary. And so this, to us, was the beginning of how we can see the epithelial cells can become transformed into cancer. And if you wait a little longer, by about 113 days on average, those tumors are now big enough we have to euthanize the animals. But we can take the tumors, we can take those little tiny tumors, and we can look at them. And using that strategy, we've been able to find several things that are different in those tumors, and they're the same things that we can find in women with ovarian cancer. The same activity that goes on there is what goes on in the early development of ovarian cancer. And so we've learned a lot from these mice. I have 3 posters that are being presented later today talking about some of the data that we have identified. But because estrogen was a risk factor, we gave some of the animals estrogen just to see what it would do. It killed the mice in half the time. So it was much more potent in a mouse than it is in humans. But using that, we identified some of the genes that are responsible for the onset, the initiation of tumor development, as well as the rate of progression, how quickly that disease will progress. And that has given us some new targets, because one of the targets we identified had never been reported in ovarian cancer in women before. It's called GREB1, long name, but GREB1 turns out to be expressed in 79% of human ovarian cancers. And we identified it because we had given estrogen to a mouse. So although the mouse model is not our favorite thing to do, we obviously want to address the cancers in women, we can learn a lot from the mouse models that will give us potential tools that can be used in women with ovarian cancer. And we also know that mice do respond to human cancer treatments. I've just finished what we call a preclinical trial for a rare form of cancer called small cell carcinoma of the ovary. It's very rare, very aggressive. It hits women in their 20s, early 20s, so it's completely different than the kinds of cancers that are most common in ovarian cancers. But we have found a treatment for this type of rare cancer. So we're very excited by that. We've just sent it in for publication, and hopefully then that will translate to a cure, or a treatment at least, for women who have this rare form of ovarian cancer. So we learned a lot. So what's next is, will the type of ovarian cancer affect which treatment will be suggested? And I'm not going to talk too much about this because this is where Dr. Herte's expertise comes in, but I will just put up this slide because this is all stuff we learned in the last decade. We knew that women had different types of ovarian cancer. If you looked at them, pathologists would look at the slide that has your tumor on it and say, oh yes, this is a serous, or this is a mucinous, or an endometrioid, or a clear cell. But we didn't know what makes the difference. What is the indicator that says this is other than looking at what the cells look like and how they're organized? That's what the pathologist was doing. But now we know the molecular events. Where are the mutations in what genes that are leading to these various categories of ovarian cancer? And so we know, for example, high-grade serous is the most common subtype of ovarian cancer, and it almost always has a mutation in P53. It is associated with mutations in BRCA1 or 2. So the women who have mutations in these genes, this is the type of cancer they're most likely to get. And so underneath each type is listed the genes that we've identified across the world for the mutations that lead to that type of cancer. That's a huge thing. It doesn't change anything at all in terms of really what you're going to be treated with, because we don't have drugs to address all of these things yet. But knowing what causes the cancer, we're halfway there, right? Now we can go ahead and figure out how to stop what is causing that cancer from growing. And let me give you one example, and I'll use the P53 mutation, but the one that has a BRCA mutation. And that's the one that leads most commonly to the high-grade serous carcinoma. So with a normal BRCA gene, remember I said that BRCA1 is important for repairing mutations, right? So in a normal BRCA1 situation, if you have a mutation, which I've drawn just by a little gap, these are strands of DNA, you've got a little mutation there. BRCA1, in common with this other protein called PARP1, together they repair that mutation. So they fix the mutation. And so that's great. If you're a cancer cell, if you're happy because your DNA has been repaired and you can live, well, that's not what we want. So we have found that if you have a BRCA1 mutation, you don't have a functional BRCA1 protein. If you add then a PARP inhibitor, like olaparib, then the combination of having a dysfunctional BRCA1 and inhibition of PARP means you end up with double-strand breaks in both DNA strands, and then you have a very unhappy cell, and that leads to cancer death. So knowing that a BRCA1 mutation needs PARP in order to repair DNA, we can take a BRCA1 mutation patient, give them a PARP inhibitor, and that actually has a much higher probability of stopping the cancer or the tumor growth in that patient. So you see, this is one example of how knowing how the mutations are that are developing these cancers, we can find a way to work around it and try to then kill the cancer cells using the strategies that are available to us. And so olaparib is now one of the treatments, and I'm sure that it will come up again in the following talks. So this was a very exciting strategy, and now we're trying to do this for all the other kinds of mutations that we know are associated with the types. And just to give you an idea of what my world looks like, so this is a cell. These are some of the pathways that exist within a cancer cell. And so there are all kinds of different pathways that all lead to controlling whether a gene is turned on or off, whether a cell will proliferate, divide, or not. On this side, we've got the cell death genes over here. Cells growth genes are up there. And so the whole thing is all networked, right? We can't sort of pull the plug on one and hope that that will be enough. We probably need to pull the plug on 2 or 3 at a time if we're going to have an impact, because the cell has amazing mechanism to take care of itself, and it desperately wants to live, especially a cancer cell. So we're going to have to hit it multiple ways. And so there's all the green or all the drugs that currently exist to target each of these pathways. I've done some of this preclinical work that has led to the trials that are available for some of these in other types of cancers and a couple for ovarian cancer. A lot of these have been tested in ovarian cancer and don't work, unfortunately. Ovarian cancer just seems to be one of the tougher ones. And so we're going to have to probably do a combination set of treatments in order to get this to work. And that's all I'm going to say, because the rest of it's going to be up to Dr. Herte. So what treatments are available to us? Well, I've shown you all kinds. We do know that surgery and chemotherapy is the standard of care for most women with ovarian cancer. And that chemotherapy is normally a combination of Taxol and platinum drugs, carboplatin or maybe cisplatin. You can have the chemo before surgery or after surgery. You can have it delivered by intravenous methods or IP chemo. You might have heard that you can have the chemo directly delivered into your abdomen. That depends on the kind of cancer and the health status of the woman. But there's lots of ways that you can play with the methods that we currently have to try to have them work optimally for each ovarian cancer patient. But clearly the consequences of chemotherapy are not ideal, and it's not specific to the cancer, right? You're killing the cancer cells, but you're also damaging a lot of the other cells, the hair follicle cells, your hair falls out, all the neuropathies. There's way too many side effects for chemotherapy. So we really do need to work on making better treatments, and Dr. Herte is going to talk about them. I'm just going to talk about one that we played with a little bit and trying to work with the Oncolytic Virus Consortium that's based in Ottawa. And this is just sort of setting the stage for one of the subsequent speakers who's going to give you all kinds of details about how immune therapies work and what's the best way. This is the brand new form of cancer treatment that is up and coming. It's very, very different than chemotherapy. It is strategies that are using your immune system to help fight the tumor in various ways. And so whether that be by vaccines, whether that be by drugs that are inhibitors to the immune system, how the immune system regulates itself, or whether you take out the T cells. Those are the cells that fight the tumor normally or fight viruses or bacteria when you have an infection. You can take those T cells out, boost them, reactivate it, identify this tumor and go after it, and then put it back in the patient and have that immune cell, the immune transfer work. But it's a exploding, let's say, area of research. Cancer immunotherapy has become a very popular area of study because there is such huge potential in this area of investigation. And so this next speakers is going to be talking about these immune therapies and what clinical trials are up and coming to apply these to ovarian cancer patients. It's coming. It's coming. So the one that I've been working on in my lab with my colleague John Bell is that if this is a cell and there's the nucleus or the genetic material, when a virus gets into a cell, it begins to multiply. That's what viruses do. And when it multiplies, it can then go into another cell, and there it will multiply again. And the advantage of the virus is that it only multiplies when it's inside of a cell. And that you can create these viruses so they are specific to multiply only in cancer cells. And so my colleague John Bell actually showed me this slide, a series of slides a long time ago, probably almost a decade ago now. And I love this so much I stole it from him, well, with his permission. So that's John Bell up in the corner. And so he had a lung cancer model, lung metastasis model, where he would take cancer cells, inject them into the vein of a mouse, in the tail of a mouse. It would go through the blood circulation and end up in the lungs, and there it would make a tumor. And so if you look at the mouse becomes very sick because it's got lung cancer now. And if you look at the lungs, this would be the normal lungs, normal healthy pink lungs. And this is what it looks like when you put cancer cells in the tail vein and they go to the lung. This is all these little nodules are obviously lung tumors. So that's not good. So the interesting thing about this strategy, though, is that if you give an oncolytic virus, it's OV up there, you give an oncolytic virus, you can get the tumor, but if you treat then the tumor with an oncolytic virus, and the oncolytic virus has been tagged with something called green fluorescent protein. So we can see where the oncolytic virus goes with the GFP. So you're looking now at the lungs, and you can see, my goodness, the virus is everywhere in the tumors in the lungs. So this is telling us that the oncolytic virus knows how to get to the tumor, and there it begins to replicate. It makes itself over and over again. It multiplies. But the interesting thing is that if you look up here, there's a little patch of normal tissue. This is still normal lung. And if you look down here, see, that's virus didn't go into the normal tissue. So this virus is training itself, or it has been trained, to only go into the cancer tissues and not into the normal tissues. And this kind of strategy, if we can get it to work on ovarian cancer, would be brilliant, because if you don't have infection in normal tissues, then all those side effects should go away that we normally get with chemo. And this is just showing a survival curve. At the start point of the treatment, the number of patients, our patients are mice, 100% of them survive. If they get no oncolytic virus treatment and you do the same thing, you put the cells in the tail and they get lung cancer, this is how long it will take them. So by 60 days, all the animals will have to be euthanized because they have lung cancer. If you give the oncolytic virus therapy, all the animals in the study survived. So it's very, very exciting. In a mouse, it works, and it works on lung cancer. And I've stolen one more from John, because this is a liver cancer patient who had a metastasis on the shoulder. And so in this case, you can actually directly inject the oncolytic virus into the tumor, right? It's easy to do because you can see it. And of course, in this case, I wouldn't be showing it otherwise, the oncolytic virus actually did reduce the tumor in that patient. And so this is all good, but there's dozens of viruses, and they've been done mostly by injecting directly into the tumor. Ovarian cancer is in the abdomen, and the tumors are everywhere, so how are we going to make that work? And that's where we stand right now, is trying to figure out how we get the viruses into the tumors and how we get the right viruses into those tumors. And Eva's going to tell you all about that. I just want one more, because I do the mouse work in advance. These are mice with ovarian tumors. This is a mouse with normal ovaries, no tumors. We just, for fun, injected them with that same oncolytic virus just to see where does it go. In the normal mouse, you can see some ended up just in the flank where the blood circulation has gone through. This is just, I think, 24 hours after injection. But if you look at the animals with tumors, you can actually see where the ovaries are. This is the GFP fluorescing. Now, they don't look like this really. We have a microscope that shows us this fluorescence. But this is where the ovaries are, the tumors are in this mice, because it's glowing. The highest intensity of virus is red. So not only do we see that it's gone largely to the tumors, but you can see that it's replicating there. And this is the other animal, so that you can actually see. It's not anywhere else. It's in the ovaries, or the tumors in those ovaries. So that's quite exciting, because we just injected it IP. We put it just in the belly just to see where the virus would go, and this is where it went. So there's promise here. I'm really excited by the possibility of oncolytic viruses, and I can't wait for Eva's talk, because I want to see what's up and coming. So I think there's potential here. I'm just going to end with the fifth category, the fifth thing that is different in the past 10 years, and that is the new resources that we have available. And I'll just highlight two. Elizabeth has already mentioned the National Ovarian Tissue Bank. This has been, I think, a remarkable achievement for all the people across Canada. These are the ones who piloted the project back in the late 1990s, 2000. Again, Nellie Auersberg, the one from Vancouver, Anne-Marie Masson, the scientist in Montreal, myself in Ottawa, and Patricia Shaw in Toronto. So these are the big 4 cities in Canada, and these are the ones that we work together to start collecting ovarian tissues for research purposes. And one of the most exciting projects we have on the go right now is called the Canadian Ovarian Experimental Unified Resource, or CUR, for those francophones, right? It's Anne-Marie's from Montreal, so it had to have a nice, soft-sounding French name. Powerful, though. CUR is your heart, for those of you who don't know French. So she is the principal investigator of this study. We have, over the last 5 years, collected 2,000 ovarian cancer cases from across the country. Samples of women who had surgery for their ovarian cancers have all come to Montreal. So that is probably the largest database of well-defined tumors, certainly in Canada, and I'm not even too sure, maybe even internationally. So it has all the tumors, but it has all the patient information as well, without the identity of the patient, but we know everything about those patients. We've been using it to look for biomarkers for ovarian cancer, something that will allow us to early detect the tumors, tell us whether you're likely to respond to chemotherapy or not, something that will give the surgeon some indication of how well you're going to survive this treatment. And primarily now, we're getting to the point where we can say, okay, if you have these biomarkers in your tumor, you should be directed into this clinical trial. And if you have these biomarkers, this is a clinical trial that's probably better for you. So that's where we are right now. This study's been going for only 5 years, and it's already being pretty remarkable in the types of outcomes it gets. And then one last slide in that, or 2 slides. Trevor Shepard's made this really interesting model system. If this is your primary ovarian tumor, the way it leaves the ovary or the fallopian tube is it actually just comes off. It doesn't metastasize like any other type of cancer. The cells just, for some reason that we don't yet know, comes off the ovary and floats around in your abdomen. But when it floats around, they actually like to stick together, so they make these what are called spheroids. And the spheroids can go anywhere. They go to your colon, they go to your liver, they go to your omentum, wherever they go. And when they land on a tissue, then they attach and begin to grow there. That's a metastasis. So what Trevor has done is he's modeled this. So there's the original, what really happens in the patient. So he's modeled this in a dish. So this is just a dish that has fluid in it that the cells will live. So he's modeled the ability of the cells to come off. They float. These cells cannot attach to this plate. And so they float. And when they float, they form spheroids. And then when he wants to, he can take these spheroids, he can put them on a dish where they will attach and do the metastasis thing. So he's finding really incredible things when cells first come free, when they form spheroids, and when they reattach. And so as soon as we understand that process, at any point we can say, okay, well, we have to stop them coming off. We need to keep them as a primary tumor and prevent them from freeing up. If we can't stop them, can we stop them from forming spheroids? Because maybe they can't survive if you can't form spheroids. And so we're looking at every step and trying to see what is going on there and see if we can at least stop metastasis if we can't stop the primary tumor. I love this model. I think Trevor's done a great job with that. And so lots of reasons for optimism. The number of ovarian cancer research in Canada has increased 20-fold in the past 20 years, from 3 in 1995 to more than 60, most of whom are in that room down the hall. We have, I believe, one of the most collaborative, cooperative networks of ovarian cancer research in the world. Canadians like to work together. We've always been team players, and I think that makes our ovarian cancer community one of the best in terms of being able to do national studies.
We are unique in our openness, and we have also developed a number of resources that will facilitate research for anyone who wants to do research in Canada. It's improved our knowledge of the disease, and I think, as I mentioned, there's some examples where we have improved capacity, so there's more of us doing the work, which means more likely we're going to come up with better strategies for screening, for early detection, and for treatment of ovarian cancer patients. And we're working on all aspects of these. I just want to show my particular crowd. We go to City Hall in Ottawa for World Ovarian Cancer Day. Those of you who aren't from Ottawa would not recognize this is the mayor of Ottawa. But these are all the folks in my lab, or some of them. And on Ovarian Cancer Canada's Walk of Hope Day, we all dress in teal. This is the seamstress in the group. She's a tech in my lab. She's also down the hall. So she made all girls teal tutus, and the guys all have teal ties. And we try to be present wherever ovarian cancer patients are to help talk to you about the research that we do and give you some indication that there's a lot going on, and we hope to offer you something that's very positive in your disease course. So that's where I'm going to stop.